I'll just wait until 6:05 to see if others are joining, and then I'll start the class. Is the screen visible? Someone please confirm if I'm audible and if the screen is visible. Yeah, I can't hear when I'm presenting the screen, so please unmute and respond if I'm asking something.
Yes.
Okay, thank you. Yeah, so I'm going to start accepting people as and when they come. So I'll just start with the session for today. Uh, so today's session is going to be a continuation of bioremediation. Whatever we saw in the previous session about bioremediation and just a second. Yeah. Okay. What is bioremediation and what are all the definitions and all that. We saw that very briefly in the last class. This is just going to be a continuation of some of the concepts of bioremediation itself. So again, identification and characterization. Uh, Even for bioremediation, this is the most important starting step because we need to understand what bacteria or what microbe we are working with. So we need to do the identification and the characterization part, and we have already seen that there are several pre-genomic approaches, which means where we don't have to do the full genome sequencing using next generation sequencing or something, and rather we can use some other techniques for identification. Some of the molecular techniques include using ribosomal rRNA, polyphasic taxonomy. Polyphasic means this involves multiple phases or multiple stages. So morphological taxonomy, and then this kind of gene sequencing based taxonomy, that is doing a particular sequencing a particular single gene and then using that to classify the organisms. FAME is a technique which means fatty acid methyl esters. The fatty acid methyl esters metabolism of the different microbes are used for classifying them, analyzing the different lipid metabolism and lipid analysis. All of these are different phases of taxonomy which can be used for classifying microbes. And the PCR based strategy, where we use PCR for sequencing different markers. So these are the molecular techniques which are pre-genomic techniques which can be used for identification of the microbes. The next thing is characterization, and characterization can either be metabolic sequencing or physiological sequencing. Metabolic sequencing just involves identifying the different pathways that are involved in the microbial energy production, energy utilization process, and substrate utilization process. So these different pathways can be characterized. And physiological characterization is what is the temperature at which it will grow at, what is the pH conditions. Finding out these different physiological conditions is the physiological characterization part of this identification. So the characterization, there are a lot of uncertainties involved, uh, because we do not know whether the particular isolate that we have isolated, isolate here means a particular strain of bacteria or a particular strain of other microorganism that is isolated from the overall environmental sample. So we'll not know how that particular isolated sample will behave under in situ conditions. In situ as in when we use that for bioremediation at a particular site in a soil or a water that is contaminated, we do not know how that particular isolate will behave. And the second thing is the fate of the contaminant. So this is the most important part of the characterization, because whatever we are processing, the contaminant should be harmless at the end of the process. So if we are modifying the contaminant but again it produces a byproduct which is again harmful, that may not be of use to us. So the most important the second most important thing is to understand the phase of the contaminant itself. So this is a small sketch of how the this technique called as FAME, fatty acid methyl esters technique, FAME technique works. So we culture the microbe from the environmental sample, and saponification process and the methylation process is conducted. And after that, saponification is addition of any agent that forms a foamy soap-like structure. So esters are formed. So this bacteria converts the substrate into esters, and that process is called saponification. Methylation is methylation of the microbe itself, and after that we extract this sample and conduct the GC analysis, and we'll be able to get this different pattern or peaks. The GC is gas chromatography. So these are volatile compounds. They'll be volatilized, and these volatile compounds can be analyzed using gas chromatography, where the mobile phase and the solvent both are gases. They move through the gases, and they move at different speeds depending on the size of the molecules itself, and we'll be able to get these peak patterns for individual microorganisms depending on how they convert the estrus. And based on that pattern, we can use a pattern recognition software to differentiate each of the microbes and classify them as different microbial organisms. So this is a very useful technique that is used for classification of microbes in a culture or in an environmental sample. So these are for classification, but in characterization these problems need to be addressed before we actually start using that microbe in any of the in situ conditions. So to understand the microbial metabolism, we need to know what substrates the microbes are converting, and what are all the byproducts. In any of the microbial metabolism, when the microbe utilizes an electron source, it will give out carbon dioxide, water, and energy, and some other byproducts, which could be nitrates and so on. So this is the basic metabolic pathway for any microbial metabolism reaction. CO2, energy, and water are produced. Uh, but we have to know what exact conditions we need to use in order to facilitate the conversion of the particular substrate that is the desired substrate, or the substrate that we want to convert. So for that, we need to understand what are all the available agents that can be used in this process. So the direct bioremediation process often happens in an aerobic condition with the help of oxygen. Oxygen is the most abundant or the most available electron source, which can help in the bioremediation process. So most of the contaminants are usually oxidized, and the water and carbon dioxide are produced from that. So oxygen is the most commonly available electron acceptor. So there should be one electron acceptor and one electron donor in this whole process. That will help facilitation of conversion of the substrate into various harmless byproducts. So this is a gradation of what is the most available to what is least available. So methanogenesis is something that happens very rarely in the environment, where carbon dioxide gets converted to methane. There are also these other processes for nitrate conversion, manganese reduction, iron reduction, and so on. So these are the different bioremediation processes. These are the different electron acceptors that can facilitate in the bioremediation process of a contaminated zone. So here we are seeing a contaminated water table, and how aerobic respiration is the direct bioremediation process that can happen. So, like I mentioned, oxygen is the most abundant and potent electron acceptor. So, here there is an example of how arsenic. So, in one of the previous examples, we have seen how arsenic is very harmful to the environment, how it causes a lot of diseases in living organisms, humans, and other living organisms, how it gets into the water table and gets inside the plants and starts contaminating the whole ecosystem, and the whole food chain gets laden with arsenic because it's a molecule that can't be easily eliminated from the environment. So oxidation of arsenic is one of the ways for degradation. So here we can see that degradation pathways are initially predicted using in silico bioremediation. So in silico bioremediation is basically just trying out different combinations by using different microbes, and giving a combination of reactions for substrate conversion, and seeing which final product is harmless for the environment. So through this, the biodegradation pathways are predicted. So arsenite oxidase is an enzyme which can oxidize arsenic and convert it into a harmless product. So this bacteria which contains arsenite oxidase is genetically modified to remove whatever is harmful in this bacteria, and retain only this property of oxidation of arsenic, and use this bacteria in wastewater treatment. So this can be this here arsenic in the soil is oxidized, and arsenic in water is reduced. and then the products are released to the environment. So this is bioremediation. So here we can, like we also saw in the previous session, whether it could be augmentation or biostimulation. So bioaugmentation is a case where we are adding extra microbes to the environment, specifically genetically modified ones, which are not originally present in the given environment, and we are externally adding them, and we are also providing nutrients, so that they convert the contaminant in the site of contamination to something that is harmless. However, biostimulation is just stimulating whatever is already existing in the soil. The native indigenous microbe is just stimulated by providing the right electron sources or nutrients, so that that microbe will now act on arsenic or whatever the target compound is, and convert it into a harmless product. So oxygen here here also for arsenic, oxygen is used, and that is the most potent electron acceptor, because in most of the reactions it's an oxidation reaction, because oxygen is very hungry for electrons, so it will readily take electrons, and it will be able to form other oxidized products. So that is why most of the biodegradation reaction also happens in the presence of oxygen under aerobic conditions. But there are also other alternate electron sources in anoxic or anaerobic conditions. Anoxic or anaerobic means the absence of oxygen itself. So here we can see different anaerobic conditions. So this is same as what we saw in the previous slide. And here, depending on what is the electron acceptor and the electron donor source, it will be either aerobic or anaerobic, or it could be phototrophic or chemotrophic and fermentation process, and so on. So these are the other different process that can happen, and for each of the process different microorganisms are associated. So for example, methanogenesis, methanospirillum is a bacteria which can produce the relevant enzyme for conversion of the waste products into methane and cellular biomass. Similarly, desulfobacterium, geobacter, azo, arcus are these different microorganisms which can produce the relevant enzymes which are required for this anaerobic conversion of the waste products. So here we also see that Rhodopseudomonas is also helpful in converting, but this is a phototrophic and an anoxic reaction. And syntrophus is another bacteria which uses some organic compounds to convert them into organic acids. So here both the electron acceptors, donors, as well as the byproducts vary depending on which alternate electron source we are taking here. And the energetics also varies depending on the free energy. Energetics also vary depending on which electron source is selected. Since I mentioned that oxygen is the most abundant and the most commonly available electron source for bioremediation, you can see that the energetics is also very feasible for the oxygen based um, bioremediation. So these are some of the alternate sources. So we'll also see individual examples for each of these different electron acceptor, and how they are converted and what product they're converting. So this is a specific example case of bacteria, Geobacter urani reducens. So here from the name itself we can say that it reduces uranium. Uranium is again another heavy element which is often generated as a waste in nuclear reactions and so on, and eliminating that is a very big problem. Here in this paper they have specifically looked at several geobacter species. So if you remember from the previous slide, geobacter species use ferric ion reduction processes, So they use electron acceptor and donor. The ferric ion access an electron acceptor in this case, and geobacter has the enzymes that can perform this reduction reaction. So they looked at several species of geobacter bacteria. Here you can see, and each of this is specific to different contaminant. So here they have conducted a DNA content based phylogenetic tree. So here they also classify them based on the electron acceptors and donors that they can act on. So this positive symbol means that they can use this particular electron acceptor or donor. So as you can see here, so this yeah, so these are for these different organisms, and they have built this phylogenetic tree based on that. So for all the geobacter species they have conducted this, and by doing this they were able to see that the sequence that they got for their organism did not combine with any of these other geobacter species, which also shows that it could possibly be a species that is unique and has a different mechanism and can act on a completely different electron acceptor and donor. And that's how they found that it can act on uranium and it can reduce uranium. So that's one example. So geobacter urani reducens specifically reduces uranium and helps in uranium bioremediation. The next example is for conversion of polychlorinated biphenyl, the remediation of this, like tetrachloroethane, tetrachloro, yeah, so yeah, trichloroethane and tetrachloroethane, and so on. So these are bioremediated by some certain bacteria. This is the bacteria that helps in uh, dehalococcicides. So this is the bacteria that helps in conversion of polychlorinated biphenyl products, and they convert them into less toxic components that are more suitable to be released back into the environment. So these are dechlorinating bacteria, and these are the only known bioorganisms so far that can help in conversion of polychlorinated biphenyl compounds without any harm to the environment. So the next thing that we are going to look at is a metal feeding microbe. So this is that microorganism, and we can see these pili like appendages. So pili is usually pili or cilia, or usually appendages that grow from the microorganism, which helps in his movement, and these usually help in moving towards the food source or the nutrient source, again, like which is specific for them. So, Geobacter metallireducens is another strain of this or species of this Geobacter species. So, earlier of this Geobacter microorganism, earlier we saw Geobacter uranireducens, but this is metallireducens. So, from the name itself, we can see that it can help in reduction of metals, heavy metals. So, here the example is for conversion of Fe3 oxide, ferric oxide, coated sediment, and manganese oxide, and it grows specifically in the presence of this conversion product itself. So it helps in the conversion of this, and it grows specifically when grown only on the insoluble forms of this ferric and manganese oxides, and they help in conversion of them into soluble components. So chemotactic means they move towards that particular component. So here we can see that they do not grow in the presence of others, and they grow only in the presence of insoluble Fe. So this is pili and flagellum, and this grows only in the presence of insoluble Fe3. Here these are insoluble Fe3, and they grow only in the presence of that. And here they are giving nitrates, and we see that this does not form pili or flagellum, and they do not move. So they start moving only towards the Fe3, and they are chemotactic. Therefore, they are chemotactic towards Fe3. So this is another specific example case where people have looked at how this microorganism will specifically grow in the presence of insoluble Fe3. Again, this is going to be like a couple of slides, and this is the last part of today's session. So this is on biodegradation. So we always see on all the polythene bags and all that, that it is degradable, compostable, biodegradable, and so on and so forth. So biodegradable, like the name says, it's something that can be degraded in the presence of a living organism. Yeah, it can be degraded only in the presence of a living organism. But before that, the most important thing to identify is point source versus non-point source contamination. So this is a major problem in understanding how to remediate contaminated environment. This is mainly also important in cases of, let's say, metropolitan waste or waste generated from homes in cities and towns. So these wastes are usually not segregated, and they are all collected together and dumped at one point. But they come from these different houses, these different sources. They contain completely different types of waste. There's glass waste, plastic waste, organic waste, human waste, so on and so forth, and all of this are mixed. All the governments are now insisting segregation at source, because let's say you're segregating waste at your house, you may be maximum generating like one bag of waste per day, and that one bag would probably have some plastic wrappers or food packaging materials. It could have fruit peels, vegetable peels, and these are probably the most or could also have some glassware or broken glassware, and sometimes also electronic waste. So this is a maximum different types of waste that can be generated in a household in a particular day. uh, so it wouldn't take much time for us to segregate that waste at the source itself, at our house itself, if we just segregate these things and keep the biodegradable ones separately, compostable ones separately. And so what is the difference between compostable and biodegradable is, composting, it can be completely converted to manure. So this is completely from biological products like fruit peels and so on, and you can do composting at your home itself with the help of earthworms and a little bit of starting culture and so on. You don't need any industrial processes for this. But biodegradable, which is where all our plastics and all of that come under, even though they're biodegradable, they are not compostable. That is, if you just throw it on the ground, the natural microbes and whatever is there in the soil will directly not convert it into something that is harmless. It requires some extra processes. It requires some specific microbes that can act on the plastic to convert it into harmless product. So that is what biodegradable is. So that is one category, and then there are non-biodegradable, non-compostable waste that only can be recycled and can't be classified in any of that. Those can be segregated at home itself before we send it out, and hence that will not result in non-point sources. So non-point sources means contamination that is coming from multiple places and combining at one source. Whereas point sources is, we can identify this is exactly where the problem is. So here, since like the cities, farms, roads, everything, all of these are non-point sources, because we don't know exactly from which house what waste came. Unless we segregate it and separately give it, there is no way of identifying which point source which waste came from. But in the contrary, factories and oil spills and all of these are point sources, because if there is a particular chemical being released from a factory, we can pinpoint and say that this chemical contaminating this water is coming from this factory. But that is not possible in case of mixed waste that comes from metropolitan households and so on. So that is the most important thing in understanding about biodegradation, and classifying as point source and non point source, so that we'll be able to understand what exactly do we need to do to contain the contamination. In case of non point sources, it becomes difficult because we don't know exactly where what is coming from and what is contaminating. So some of the common point source discharge of pollutants to waters in the United States. So like municipal waste. So these are so these again can be even though this comes, so these things, even though it is a combination from multiple other houses and so on, at least we know that all of this water came from this stormwater drainage system, or the sewer system, or construction water system, or whether it is from moving vessels, that is ships that are moving, or is it from a processing plant or an industry or so on. So if wastewater comes to a particular region, we'll be able to identify, okay, this comes from this city, this wastewater is from here. But this is not in case of solid waste, this is only for liquid waste, we'll be able to identify this. But like I said, in case of solid waste, it becomes a non point source, because solid waste is usually mixed waste, and unlike the water waste. So for this we need again a specific mechanism, so that we can effectively address each of these point source and non-point source problems. So especially for non-point source problems, if we do waste segregation at source, and if we identify biodegradable products and keep them separately, we can employ these different mechanisms for degradation processes. So here this is a mechanism that is proposed for degradation of benzene. Benzene is an aromatic molecule that is a contaminant that is there because of many of the industrial contaminants. And pseudomonas, again, pseudomonas is like we know is the super bug which can convert several microorganisms. So here they are using multiple microorganisms, because each of the byproduct has to be converted by the enzymes that are present in different microorganisms. So microbes use enzymes for the biodegradation process, and the most important thing is to identify which microorganism produces what enzyme. So benzene is converted to phenol by pseudomonas, and another strain of pseudomonas can again convert phenol to catechol and hydroquinone. So now catechol and hydroquinone are not degradable by pseudomonas, and we have to employ a different microbe here. So it can be acetogenic bacteria, which will convert catechol and hydrocolin into benzoquinone. And again, acetogenic bacteria. So acetogenic is something that converts some of these cyclic compounds, and that will convert it into malic acid. After it forms malic acid, methanogenic bacteria can convert it into oxalic acid, and further to carbon dioxide and water. So our final aim for biodegradation is to convert it into carbon dioxide and water, which is abundantly available in the environment and not harmful. So to achieve this, we should be able to understand what are all the different byproducts that comes in each of these processes, so that we can use the respective yeah so that we can use the respective microorganism for each of these conversions. Here that is the pathway for degradation of benzene by a combination of these different bacteria. Actually, that is all I have for today. So if there are any questions, if people who join late, uh, if you guys want, if you guys want me to repeat some of these things, um, you guys can go ahead and ask your questions. anyone who joined new wants any of this to be repeated, because it will be there in the recording. You can always go back and check the recording and see if there's something you don't understand. You can always check that.
Where we can find these recordings?
So in the NPTEL page itself, they are embedded. So maybe I can show that in today's session. Um, I don't know. Let me just open and share my screen. Because I've been told that it is there, but maybe it's better I just show it once during the session itself.
Once we log in, we'll find it out in our session. So every week there will be a reminder as well as even for the week session, and then the yeah, the session will be there. Problem solving.
Yeah, so I'll just share my screen and yeah, is my screen visible? Are you able to see the NPTEL login?
Yeah, now it's visible.
Yeah, so here you just go to the course. So this is your course portal. You'll have these different weeks content here, and you'll also have this link to these sessions. And every week there, like sir mentioned, there's every week there's a reminder that they send, also about the content, everything that they include these things as well. But if you go down towards the bottom, there's this tab called problem solving session, which is specifically for these 1 hour sessions that I conduct. So you click on this, and here you click here. It opens another tab. So I'm not sure if that tab is visible. Let me just, yeah. So once you click here, it goes to this Excel sheet where you have all this every week's links. So there's a YouTube link where I upload the entire recording of the video to YouTube. So you can open this and check the whole video there. Uh, so this is also the presentation folder. So all the presentation slide decks that I have, everything is uploaded into this one folder. You can open and see individual folders. So this is also a backup video in case you want to download this video and keep this for yourself. You can also open this link, and you can see the recording of the video here as well. So this is where all that is embedded. And you also have a survey on these problem solving sessions, so you can go and fill this survey using this link and see if there is any specific thing that needs to be included in these problem solving sessions. If there are any questions and doubts that you have, that you don't understand about a particular session, you can always come and ask about that, and I will include those things in my slide deck as well. I hope that's clear now.
If we have to ask doubts, what was the thing you told finally? I couldn't get that.
So there is a way to give feedback, but you can also send me the questions directly. You can ask the questions here during these sessions. You can ask your questions if you have any question from that particular week or even from the previous weeks, and if you have missed out, you can ask that question here during these 1 hour sessions. Or you can also write to me in my mail ID. I will once again put my mail uh, ID here so that you guys can mail me in case you have any questions. Yeah, that's my mail ID. You can send your queries during the course of the week while learning. Also, if you have any queries, you can always send that query to me. If it's any query that is related to something is wrongly updated in the answers or something like that, the NPTEL team will obviously look into it and they'll correct it. But if it is something conceptual, you're not understanding a particular concept, definitely try to address that in these sessions. So yeah, I got a mail 3 days ago from Hitesh in the discussion panel saying that he has a doubt in assignment 8 where the solution for question number 9 is not matching the information given in the slide. So please use only the slides as the information. Those are the correct information. Sometimes there might be mistakes in the final solution, the assignment solutions folder. So please always look at the slides. If there are things that needs to be corrected, NPTEL team will definitely correct these things and send a revised mail. Even for one of the previous weeks also there was an issue with the answers and they corrected and they sent it back. So please refer only to the lecture course material for referring to the answers. Is that clear, or are there any other questions? Are there any questions? Any clarifications that are required? So today is the ninth session. We still have three more sessions to go, and after that, you will have an exam. So you can start reviewing from the initial session, and if you have any doubts from the initial sessions, also you can ask here so that before the exam, you're clear and you have an understanding of how to approach the exam and so on. All right, so if there are no more questions from today's session, I think a lot of people joined uh, like a little late. So please do go over the slides, the videos that I'll be posting soon for this session, and feel free to write to me in my mail ID, or you can ask these questions in the next session also if you have any queries. Thank you so much for attending today's session.